Hi, my name is Sirui Chen, and I'm a research student who works in Dr. Melanie Berkman's lab at Suffolk University. Today, I'm going to talk to you about our project on the localization of two interacting proteins, components of the conjugation machinery of Bacillus subtilis. In our lab, we study the bacterial conjugation, which is a process that involves transferring DNA from a donor cell to a recipient cell through a translocation channel. We call those translocations channel the conjugation machinery. In real world, the conjugation is relevant since it can spread genes involving virulence, metabolism, and most importantly, antibiotic resistance. There are many proteins that are involved in the conjugation machinery of Bacillus. Our study mainly focuses on two of them, ConB and ConE. In panel 2, we show you the model of predicted topology of ConB and ConE. ConB consists of 4 major domains which are located throughout the cell, whereas ConE is only cytoplasmic. So we are curious to know the subcellular localization of ConE and whether the localization depends on other proteins since it lacks a transmembrane segment. Previously, our lab has found that ConE-GFP localizes to the cell membrane, predominantly as cell poles. The localization is also ConB-dependent, as shown here on the left. The wild-type ConE-GFP is able to localize to cell poles. Next, I constructed a ConE strain with deletion of ConB gene. On the right, we see the ConE-GFP in absence of ConB genes localized throughout the cytoplasm. So our next question is, where does ConB localize? And does the localization depend on ConE or other conjugation proteins? To answer this question, I first observed the wild-type ConB fused with mCHERRY. We saw that ConB-mCHERRY localization is similar to ConE-GFP, which is primarily at a cell pool. Next, I constructed a strain of ConB with deletion of all conjugation genes. We saw that the polar localization of ConB is still preserved. On the right, I constructed a ConB strain with deletion of its transmembrane segment. We observed that ConB-mCHERRY localized throughout the cytoplasm. Thus, we conclude that the localization of ConB is independent of other conjugation proteins, but requires its own transmembrane segment. This brings us to our last question. Which domain of ConB are required for ConE localization? So in panel 5, I constructed 4 ConE strains with each lacking one of the 4 ConB domains. Here, we saw that wild-type ConE-GFP was fully ConB gene localized at cell membrane, where in absence of N-terminal tail, transmembrane segment, and central NTF2 domains, ConE-GFP mislocalized throughout the cytoplasm. C-terminal NTF2 domains, on the other hand, is somewhat not required since polar localization is partially observed. To summarize, ConB and ConE are 2 crucial proteins of conjugation machinery in Bacillus. ConB and ConE both localize in a similar fashion, where the localization of ConB is independent and the localization of ConE depends on ConB. 3 out of the 4 domains of ConB are required for ConE localization. Our work provides new insight onto the subcellular localization of 2 conserved conjugation proteins. Finally, I would like to thank NSF and Suffolk University for supporting this project. Also, thank you for listening.